Welcome to MedTech ISO. I am your host Arvind Rathore, and in today's video, we will focus on one of the most essential aspects of ISO 109931, toxicological risk assessment. This process is at the heart of ensuring the biological safety of medical devices as it evaluates potential toxic risks associated with the materials and the chemicals used. Whether you are a seasoned professional or new to this field, this video will provide a detailed step-by-step guide on conducting a toxicological risk assessment, highlight the common pitfalls, and share actionable tips to avoid them. Let us dive in. What is a toxicological risk assessment? At its core, a toxicological risk assessment under ISO 109931 is a systemic evaluation of whether any materials, chemicals, or byproducts in your medical device could pose a toxicological risk to patients. This process doesn't just stop at identifying risk, it also determines whether the level of exposure to potential toxins are acceptable or require mitigation. It is about ensuring that the device remains safe under all foreseeable conditions of use, whether it is a short term contact device like surgical instrument or a long term implant like hip joint. Toxicological risk assessment is critical because it provides the scientific basis for ensuring that devices meet regulatory safety requirements. It is not just about passing tests, it is about understanding the device at a molecular level to predict and prevent adverse effects. The step 1 is identifying potential sources of toxicological risk. The first step in any toxicological risk assessment is to identify all potential sources of exposure. This means you need to consider every material, chemical, and byproduct involved in the device's life cycle. Start by evaluating the raw materials used in the device's construction, including polymers, metals, adhesive, and coatings. Then move on to assess any chemical introduced during manufacturing processes such as cleaning agents, lubricants, or sterilization residues. For example, a catheter might appear simple, but it could introduce risk from the base polymer, a coating designed to enhance performance, or even residues left behind by the ethylene oxide sterilization process. Each of these components needs to be scrutinized. Additionally, don't overlook degradation products. Materials in contact with body fluid can degrade over time, releasing new chemicals that may not have been present initially. For example, a polymer used in an implant might degrade into the smaller molecules that could trigger an immune response. These potential degradation products should be included in your analysis. The step 2 is chemical characterization. Once you have identified potential sources, the next step is chemical characterization. This involves using advanced analytical techniques to detect and quantify any chemical that could leach from the device. The goal of chemical characterization is to create a detailed profile of all extractables and leachables from the device. Extractables are the substances that can be extracted from the device under exaggerated conditions, while leachables are substances that leach out under normal use. Both are critical to understanding the device's toxicological risk. The step 3 is toxicological profiling. Once you have identified the chemicals, the next step is to assess their toxicological profile. This involves evaluating each substance's potential to cause harm based on existing toxicological data. Toxicologists used databases, published scientific literature, and regulatory guidelines to gather information on the chemical's effects, such as potential for cytotoxicity, does it kill cells? Genotoxicity, could it damage DNA or cause mutations? Carcinogenicity, does it increase the risk of cancer? Reproductive toxicity, could it harm reproductive health? Systemic toxicity, could it harm internal organs or systems? If a substance lacks sufficient toxicological data, you may need to conduct additional tests to fill the gaps. The concept of threshold of toxicological concern (TTC) is particularly useful there. TTC establishes safe exposure limits for trace chemicals even in the absence of extensive toxicology data. Step 4 is risk evaluation and mitigation. Risk evaluation is where you determine whether the levels of identified substances are acceptable. This involves comparing the exposure levels from your device to established safety thresholds. If a substance exceeds its safety threshold, it is considered a risk and must be addressed. For example, if a chemical leased from an implantable device is found to exceed the TTC for systemic toxicity, you may need to take corrective actions such as switching on to an alternative material, modifying the manufacturing process to reduce residual chemicals, adding protective coatings to minimize exposure. Risk mitigation isn't just about fixing problems, it is about designing a safer, more robust device from the ground up. Step 5: Common pitfalls in toxicological risk assessment. Now, let us discuss some common pitfalls and how to avoid them. Number 1 is incomplete material characterization. Many manufacturers skip or inadequately perform this step, leading to undetected risk. To avoid this, ensure a thorough analysis of every material, additive, and byproduct. Number 2 is neglecting degradation products. Some focus solely on the initial material and overlook degradation byproducts, which can introduce new risk. Always include accelerated aging studies to evaluate long term effects. Number 3 is over reliance on generic data. Using generic toxicologic data for materials without considering the specific context of your device can lead to inaccurate assessments. Tailor the evaluation to your device's intended use. Number 4 is insufficient documentation. Poorly documented toxicological assessments can delay regulatory approval, even if the device is otherwise safe. Maintain detailed record of every step, including justifications for safety thresholds and risk mitigation measures. Step 6 is documentation. The last but perhaps the most critical step is creating a comprehensive toxicological risk assessment report. This document brings together all the data from material characterization, chemical profiling, toxicological evaluation, and risk mitigation strategies. It must clearly outline the rationale for your conclusions and demonstrate that all potential risks have been identified and addressed. Regulatory bodies like the FDA and EU MDR place significant emphasis on the quality of this report. A well prepared report not only facilitates approval but also showcases your company's commitment to safety and compliance. To summarize, conducting a toxicological risk assessment under ISO 109931 involves: number 1, identifying potential sources of exposure; number 2, performing chemical characterization; number 3, profiling the toxicological risks of identified substances; number 4, evaluating and mitigating risk; number 5, compiling a detailed toxicological risk assessment report. Toxicological risk assessment is a cornerstone of ISO 109931 compliance. By investing in a thorough, detailed process, you are not only meeting regulatory requirements but also ensuring that your device is safe, effective, and trusted by patients and healthcare professionals alike. If you found this video informative, please like, share, and subscribe to MedTech ISO. Have questions about your specific toxicological risk assessment? Drop them in the comment below and I will be happy to guide you. Thank you for watching, and I will see you in the next video as we continue exploring ISO standards and medical device safety.